And so we are going to turn the seminar over to Dr. Jay Grobler from Merck. So Dr. Grobler is Executive Director of Infectious Diseases and Vaccine Discovery at Preclinical and Early Development at the Merck Research Laboratories. He will be speaking on Molnupiravir, a broad-spectrum antiviral for the treatment of COVID. Jay currently leads early clinical development for infectious disease programs and oversees biology for pipeline and marketed infectious disease products at Merck. Prior to this role, Dr. Grobler had leadership roles in discovery and early clinical development, focused on identifying new agents for treatment of infectious diseases, including HIV and HCV. Among his contributions, Dr. Grobler carried out seminal studies on the mechanism of action of HIV integrase inhibitors, which contributed to the identification of Infectious. More recently, he led the preclinical and early clinical development of islatravir, first-in-class non-nuke reverse transcriptase translocation inhibitor. And he received his undergraduate and graduate degrees in biochemistry and molecular biology from the University of Miami in Florida, and subsequently completed training as an IRTA fellow at the National Institutes of Health before joining Merck in 1997. So I'll turn this over now to Jay.
Great, thank you, and hello everybody. I cannot seem to advance my slides. Thank you. So Molnupiravir, shown on the right, is an orally administered prodrug of N-hydroxycytidine, a nucleoside analog with broad-spectrum antiviral activity against RNA viruses, including SARS-CoV-2. It acts by a mechanism called viral error catastrophe that I will describe shortly. A phase 3 clinical trial evaluating the efficacy and safety of Molnupiravir for the treatment of non-hospitalized patients with COVID-19 is currently ongoing. Molnupiravir has a long and interesting history. The history starts with the discovery of N-hydroxycytidine that was first described over 50 years ago. Over the past couple of decades, N-hydroxycytidine, which I will refer to as NHC, has been shown to have antiviral activity against RNA virus pathogens, including those that infect humans. These include, for example, the hepatitis C virus, influenza virus, and other coronaviruses. Given its spectrum of activity against all of these viruses, it looked like a very attractive broad-spectrum antiviral drug. The challenge with NHC, though, was that it had low oral bioavailability in some preclinical animal models and was predicted to have low oral bioavailability in people. What that means is that when administered orally, by a pill, for example, it was not projected to be taken up into circulation in high enough amounts to elicit meaningful antiviral effects. A breakthrough came a few years ago when scientists at Emory University made what is called a prodrug of NHC. A prodrug is a compound that can be metabolized by the body to produce a drug. They refer to this molecule as EIDD 2801. It is now what we call Molnupiravir or MK4482. The prodrug moiety appended to NHC enables it to be absorbed and then rapidly converted to NHC by the body, and thereby making it orally bioavailable. Oral bioavailability greatly simplifies dosing. One can take pills at home, and it allows for simpler and more rapid treatment of a larger number of people earlier in infection. With this discovery, a company called Ridgeback Bio had licensed the drug from Emory, still called EIDD 2801 at that time, and were planning to test it in clinical studies against influenza, one of the viruses against which the drug is active. That is, until the SARS-CoV-2 pandemic hit. Obviously, given its broad-spectrum antiviral activity, it was quickly tested and shown to inhibit SARS-CoV-2. And soon after, we at Merck entered a partnership with Ridgeback Bio to test in clinical trials for the treatment of COVID-19. As mentioned, Molnupiravir is a prodrug of NHC that enables its oral administration. Molnupiravir itself does not circulate in the body. It is rapidly metabolized to release the nucleoside NHC into circulation. Like other nucleosides, NHC must be taken up by cells and converted to NHC triphosphate, the anabolite that has the activity against the virus. Structurally, NHC is very closely related to the natural nucleoside cytidine and differs only by the presence of a hydroxyl group at the N4 position. This simple substitution results in what is called tautomerization and allows the base to interconvert between two forms, oxime, which is the favored form, or hydroxylamine. These tautomers, which interconvert and exist in equilibrium, look very similar, differing only in the position of a double bond with either of two nitrogen atoms. But they behave very differently with regard to how they base pair with other nucleotides. Both the oxime and hydroxylamine forms are competitive substrates for the viral RNA polymerase. As the hydroxylamine tautomer, NHC triphosphate is recognized and used in place of CTP, while as the oxime, it behaves as UTP. While cytidine will only pair with guanosine, the presence of the hydroxyl group in NHC allows it now to base pair with either guanosine or adenosine, depending on which tautomeric state it's in. During viral replication, NHC gets incorporated not only opposite G as with CTP, but opposite either G or A, depending on whether it is in the hydroxylamine or oxime tautomeric states. Once in the viral genome, it can then template for incorporation of either G or A. This promiscuity in base pairing leads to the accumulation of errors within the viral genome, which ultimately results in the inability of the virus to replicate. This mechanism is referred to as viral error catastrophe. I mentioned earlier that NHC shows activity against a range of RNA viruses that are important to human disease. Some of the viruses against which NHC has been evaluated are shown here. This list includes commonly observed viruses such as flu and RSV, as well as some that are associated with episodic outbreaks. Potency is more or less similar across these pathogens, and where evaluated, it has been shown to act by inducing viral error catastrophe. While Molnupiravir is currently only in clinical trials for treatment of COVID-19, given the urgency of the pandemic, this profile suggests the potential use for this molecule more broadly to treat other RNA virus infections. The viral genome is the instruction manual that the virus uses to make new progeny viruses. Viral error catastrophe is essentially changing the instruction manual to the extent that new viruses can't be made, or if made, are defective to the extent that they cannot infect new cells or replicate. We can measure all of these things when characterizing the effect of NHC on viruses. Sequencing of the virus following treatment demonstrates an increase in the number of viral genome mutations consistent with the mechanism of action. Infectivity assays demonstrate a profound decrease in the ability of the viruses to infect new cells or replicate. And viral RNA assays demonstrate the decrease in virus produced due to treatment with NHC and the induction of viral error catastrophe. Antiviral activity of NHC against SARS-CoV-2 and demonstration that inhibition results from viral error catastrophe come from both in vitro and in vivo studies. In vitro, NHC exhibits potent activity against SARS-CoV-2 with IC50s ranging from subnanomolar to low micromolar, submicromolar to low micromolar, depending on the assay methods and the cell lines used to perform the studies. Here, for example, are data from a paper by Tim Sheehan and colleagues published last year in Science Translational Medicine. They reported an IC50 of NHC against SARS-CoV-2 of 300 nanomolar in viro cells. They also evaluated antiviral activity in CALO3 cells and in human airway epithelial cells, where they measured infectious virus production. Those data are shown here. In both CALO3 cells and in human airway epithelial cells, there was a dose-dependent reduction in virus titers. And in CALO3 cells, the IC50 reported was 80 nanomolar. In human airway epithelial cells, a multiple log drop in virus production was observed. Similar results were obtained when NHC was tested against SARS-CoV-1 and MERS, where a several log drop in infectious titers was observed, as shown in the top panels, with similar trends observed in copies of viral genomic and subgenomic RNA, shown in the bottom panels. Furthermore, they demonstrated that the antiviral activity of NHC is associated with increased viral mutation rates, consistent with the mechanism of action. For the study shown here, they used MERS and demonstrated that the error rate was significantly increased with NHC treatment. Remdesivir, which acts as a chain terminator, did not increase the error rate despite similarly reducing virus titers in a dose-dependent manner. With direct-acting antiviral drugs, the potential for the development of resistance is always a concern. Development of resistance is a selection process. Viral RNA polymerases are naturally error-prone, and copying of viral RNA is imperfect, and occasionally mutations are introduced during replication. Resistance develops when these mutations allow the mutated virus to grow better in the presence of drug than the virus from which it was derived. The potential for the development of resistance can be predicted by conducting resistance selection experiments in vitro, where the virus is grown in the presence of increasing amounts of drug. Shown here are data from a publication by Agostini and colleagues, where they reported the results of resistance selection experiments with NHC, with the mouse hepatitis virus, and with MERS. After 30 passages in the presence of increasing NHC concentrations, neither virus developed robust resistance to NHC. Similar results have been reported for other viruses and support the conclusion that NHC has a high barrier to the development of resistance. The antiviral activity of Molnupiravir has been characterized in animal studies against several viruses, including SARS-CoV-2 and other coronaviruses. These studies more or less take the same form and entail infecting animals with the virus of interest, starting treatment, and then monitoring for the virus and the development of disease signs. In some cases, treatment can be initiated before infection to determine the prophylactic efficacy of the drug. In other words, can the drug prevent infection? Molnupiravir has shown both prophylactic and treatment efficacy across the animal models and range of viruses against which it has been tested. Here I will highlight just a couple to illustrate the point. For example, Wall and colleagues recently demonstrated prevention and treatment of SARS-CoV-2 infection in a mouse model with implanted human lung tissue. In this study, they dosed some animals 12 hours before challenge, some received no drug. Those that did not receive the drug became viremic and showed evidence of infection, while those that received the drug were fully protected. In another study by Sheehan et al., they demonstrated that early treatment of SARS-CoV-1 infected mice prevented body weight loss, reduced viral loads in the lungs, and abrogated loss of pulmonary function. And finally, in another study, Cox et al. Demonstrated that treatment of SARS-CoV-2 infected ferrets with Molnupiravir twice a day significantly reduced SARS-CoV-2 load in the upper respiratory tract. In this study, they made the observation that untreated SARS-CoV-2 infected ferrets can infect co-housed ferrets. Remarkably, however, treatment of the infected ferrets completely suppressed spread of the virus to untreated contact animals. These results, along with the pre-exposure prophylaxis described by others, suggest that Molnupiravir, used either pre or post-exposure, has the potential to break SARS-CoV-2 community transmission chains. So as part of Merck's comprehensive non-clinical program to characterize the safety profile of Molnupiravir, two distinct rodent mutagenicity assays were included. These assays, Big Blue and Pig A, provide a measure of an agent's potential to cause changes in DNA in whole animals. Animals were administered Molnupiravir for longer and at higher doses than those employed in human studies. The totality of the data from these studies indicates that Molnupiravir is not mutagenic or genotoxic in vivo mammalian systems. Last year, Merck started two randomized, double-blind phase 2-3 clinical trials of Molnupiravir versus placebo. These studies called Move In and Move Out. These were for the treatment of COVID-19. Move In is an inpatient study in adults hospitalized with moderate or severe COVID-19 and symptom duration of less than or equal to 10 days. The endpoint of the study was time to sustained recovery. Move Out is an outpatient study in symptomatic outpatient adults with symptom duration of less than or equal to 7 days. The endpoint of this study was hospitalization or death. Both studies interrogated doses of 200, 400, or 800 milligrams administered BID for 5 days versus placebo. And both studies have now completed enrollment of phase 2 earlier this year. In both studies, all Molnupiravir doses were generally well tolerated. No safety signals of concern were observed, and no dose-limiting toxicity was observed at the highest dose of 800 milligrams. Consistent with Molnupiravir's mechanism of action, an increased SARS-CoV-2 mutation rate is observed at all doses compared to placebo. Infectivity data show faster time to replication negative virus on Molnupiravir versus placebo, and RNA data show a faster and more profound reduction in viral load on Molnupiravir versus placebo by various measures, including change from baseline compared to placebo, the slope of the decline in viremia, and the proportion below the limit of quantitation. Regarding efficacy, in Move In, there was not a clear dose effect on sustained recovery, and given the limited potential benefit, we will not be initiating phase 3 in this population. In Move Out, the outpatient study, there were fewer participants in the Molnupiravir treatment groups that were hospitalized or died versus placebo. The number of events reported are not sufficient to provide a meaningful measure of clinical effect. However, benefit shown in subgroup analyses supports potential benefit for treatment. This outpatient study has moved forward to phase 3 with a focus on patients early in the course of disease and those with risk factors, including men and women greater than 60 years of age. So if nothing else, I hope what you take away from my presentation today is that Molnupiravir is an orally administered broad-spectrum antiviral drug that acts by the mechanism of viral error catastrophe and which has shown proof of concept in preclinical animal models against multiple RNA virus pathogens associated with human disease, including SARS-CoV-2. Molnupiravir has demonstrated a virologic response to treatment in patients infected with SARS-CoV-2 with no observed safety signals in a manner consistent with its mechanism of action, and that Molnupiravir is currently in a phase 3 trial to study the efficacy and safety of Molnupiravir for the treatment of non-hospitalized patients with COVID-19. And with that, I would like to acknowledge the work done by scientists at Emory who came up with Molnupiravir, our partners at Ridgeback Bio, and my colleagues at Merck who are working to advance it through clinical studies, and the participants in our clinical trials. I would also like to thank you for your attention, and I am happy to take questions.
Thanks so much, Jay. This was fantastic talk. And actually, a lot of the questions that were coming in, you subsequently answered in the talk. So I have just a couple of questions. So the first one is there a plan to do a directed evolution to identify resistance mechanisms using an in vitro assay and other variant libraries in order to provide some predictive power to identify those strains that may escape the molnupiravir activity?
So it's been really tough to identify things that escape molnupiravir antiviral activity so far, just given its spectrum of activity and its structural similarity to the natural nucleoside. I don't think there are easy handles to discriminate against it. Obviously, we and others continue to intensively interrogate this question. It's quite important. I didn't go into detail on the resistance selection experiments that have been done, but if you look at the viruses that come out at the end of those selection experiments, they are loaded with the mutations that you might expect as a result of treatment with NHC, but you don't really see you don't see any shifts in potency. So it's not a failure to get mutations; it's a failure to get mutations that impact the biological activity.
Great, thank you. And is there any interest in challenge studies using 229E or OC43 or other human coronaviruses? And is there any effect of the drug on alpha coronaviruses?
I think the question about challenge studies might be best positioned for somebody in our clinical group. With regard to spectrum of activity, I think across the coronaviruses that we've looked at, there has been comparable activity. Was that your question, or?
Okay, thank you. Yeah. Yeah, okay. Thanks very much.